Good morning and welcome to Emerald Ash Borer University. This is Robin Usborne coming to you from Michigan State University. My EAB University colleagues are Professors Cliff Sadoff and Elizabeth Barnes from Purdue University and Amy Stone from Ohio State University Extension. Today we are happy to share with you the webinar entitled Detecting and Monitoring Invasive and Non-Invasive Species from Neon Pitfall Traps, presented by Mike Weiser. Mike is a research associate in the Geographical Ecology Group in the Department of Biology at the University of Oklahoma. He did his master's at Oklahoma with Mike Kaspari and his PhD at the University of Arizona with Brian Enquist. Mike's research program uses field collection and biodiversity informatics to describe patterns of and test hypotheses about biological diversity across spatial and taxonomic scales. Mike collaborates broadly and has published more than 60 papers on taxa ranging from soil microbes to ants to trees, and one that has all three. And as a side note, Mike says his office is officially rated as a tornado shelter by the University of Oklahoma. To our participants today, we welcome your questions and comments. Please type them in the Q&A feature or the chat pod, and we will respond to them after the webinar presentation. Tomorrow, you will be emailed a link to a short, voluntary, confidential survey that we hope you will take the time to fill out. It helps us bring these free educational webinars to you. The email will also include our presenters' contact information if you have questions and comments after the webinar is over. For those of you wanting CEUs, this email you will receive is especially important, as it will give you information on how to obtain CEUs for viewing this live webinar. This webinar is being recorded and will be available for viewing later on the EAB University page of the www.emeraldashborer.info website. Thank you all for attending today, and Mike, please unmute your microphone and you can begin your presentation.
Thank you very much. So I'm Mike Weiser. I think everything important was said in the introduction, but I'm going to talk with you today about some of the methods that we've developed and hopefully some of the downstream things that we're going to be able to do with these methods. So all of what I'm presenting today comes from a collaborative macro systems grant that was given to the five of us. Mike Kaspari, Cam Seiler, and I are at the University of Oklahoma. Matt Miller is starting a job in conservation genomics in the United Arab Emirates. And Katie Marshall is a physiologist at the University of British Columbia. But I should also mention the people that are actually doing the work. These are the Sierra Smith, Jess McLaughlin, Tanner Ordery, Miranda Theriot, and Jarrett Blair are all graduate students and one's an undergrad that have worked to develop these methods and to actually implement them. So I'm going to start by talking about the stuff that I'm more interested in, which is large-scale patterns of biodiversity. And more particularly, I want you to think about how that data is collected. Then I'll talk a little bit about NEON. I'll describe the pipeline, show a tiny bit about the diversity gradients, and then I will get to the stuff that you guys want to hear about. So when developing a large-scale biodiversity database, it takes a lot of time and effort. When you're looking across taxa, you have to go out, you have to observe or sample those taxa, take them back to the lab, identify them. And that leads to kind of a taxonomic limitation for a lot of biodiversity data sets. I'm trained in ant ecology, so if I had to identify my own samples, I'm pretty much limited to ants. Maybe I could do trees, at least in North America, north of Mexico. And since you're actually putting out this effort, the biodiversity data sets can be spatially limited when you're collecting the data yourself. So in a lot of cases, what happens is people are using other people's data and they're using interpolation or modeling or limits in order to say something about the spaces between the observations. And kind of by necessity, the way that a lot of this is gleaned, if you're working at very large scales, it's difficult to get standardized data sets across taxa and even within taxa. And so my interests lie in looking at large-scale patterns of biodiversity. Now, this graph on the right, we're not going to get too much into it, but the open circles are basically the species richness of New World woody plants from the southern tip of South America to as far north as woody plants go in Canada. So this work, I'm only mentioning these three examples so that we can think about how much time and effort it went in to create these data sets. So this data set includes 13,000 species of woody plants from more than 1.3 million individual observations. I collated these data basically by getting museum references and literature references that had geospatial locations on them. And then I interpolated the range extent of the woody plants to generate this latitudinal gradient in species diversity. Another paper that I worked on involved taking the list of species from about 250 different places. So these are all vascular plants that occur at a given place. Now, the generation of each one of those published flora involves thousands of hours of sampling and identification just to make the species list. Another example that I participated in was led by Benoit Guinard, who's at the University of Hong Kong now, but he basically databased the entire ant literature. Now, I say approximately the entire ant literature because I believe they still have some Russian publications that have not been digitized and they could not get their hands on. But this is ant species richness by political divisions. And so this is the amount of effort that it went into make this map if you sum across all of the collections that were made and published in the literature, literally tens, if not hundreds of thousands of hours to get this data. So enter a nationally standardized way of collecting ecological data. NEON, or the National Ecological Observatory Network, has 81 sites total, but it has 49 terrestrial sites that are located, all of the dots on this map. So they're in Hawaii, Alaska, Puerto Rico, and then distributed across the continental United States. So NEON collects all types of data, but we're only going to talk about one form of the data that they collect. They study many sentinel taxa, including vertebrates, but the invertebrate arthropods that they collect mosquitoes using CO2 traps. They collect ticks by dragging sheets across the ground. But of interest to us is that they collect ground beetles using pitfall traps. So in case you've never seen a pitfall trap, it's a cup that's buried in the ground so that it's flush with the soil surface, and it's filled with a preservative that well, they use propylene glycol, which is ostensibly odorless. Organisms will crawl across the ground and fall into the cup. NEON leaves the cups out for 2 weeks, which is a very long time to leave pitfall traps out, in my opinion. But they're collecting over the entire growing season at the sites that they collect pitfall traps at. They have arrays of 4 pitfall traps, and they have 10 arrays per site. And so the arrays are set up at this kind of spatial scale. The data that we're going to be talking about today comes from 2016. And in 2016, NEON was collecting 4 pitfall traps. But this is their current setup where they only do 3 pitfall traps, hoping to reduce the amount of effort by 25%. And they're distributed across the landscape at kind of a more regional scale. So the kind of darker beige area are the limits of the Jornada LTER in southern New Mexico. And so I don't know which one of these plots has the pitfall traps, but 10 of these plots in here have pitfall trap arrays and are distributed somewhere within 46 square kilometers. And here's kind of just zooming out a little bit more. The red at the bottom would be the north end of Las Cruces, New Mexico, just to kind of give you a scale at which they're collecting. So they're leaving out the pitfall traps for 2 weeks, and then they take them back to the lab. They remove the adult carabid beetles, and they leave what they call invertebrate bycatch, which they then archive in 50 milliliter centrifuge tubes with 95% ethanol. So here is an example of a couple of trap events from Kanza LTR in Kansas, where, I don't know, big Passymachus beetles that are being collected. So they have this incredible resource that is being collected in a standardized fashion every summer for approximately the next 30 years. And so they want to have this resource exist over the length of time. And so you can ask to destructively sample, but at the scale that we're trying to do our work, we're not allowed to destructively sample these pitfall traps. So I'm going to introduce the pipeline that we use to get diversity and abundance of the invertebrate bycatch from the neon samples. It involves what I would consider 3 different pipelines. Excuse me. The first is the Metagenomics Pipeline, where we take the 50 mil centrifuge tube and attach it to a filter and then vacuum the ethanol across that filter. So we're taking the liquid that the arthropods or the invertebrates are sitting in, and we're filtering that and then returning it to the tube. We then take the filter out and cut it in half. This is a figure stolen from the paper cited in the bottom right. And then we extract the DNA from the filter. Well, we cut the filter in 3 pieces and put 2 in the freezer, and then extract DNA from 1/3 of the filter. And then we amplify, for what we're going to talk about today, these are the primers that we use. We amplify the DNA and we label it with a particular label that allows us to know what sample it comes from. And then sequence, then we use metagenomic sequencing to get the sequence from the DNA. Now, the second step of this is so the output of the sequence is a file that just has, sequence 1 to sequence 15 million or how many ever come out. We take those sequences and we use just standard bioinformatics methods to group into operational taxonomic units, which we then take those consensus sequences and the OTUs, and we extract a taxonomy from a blast search, giving us kind of a species list for an event that occurred. So this is the actual species list for the event that occurred at the 3rd array of Delta Junction, which is in Alaska, in the year 2016 on July 20th. We can then, for the data that we're talking about today, we took one trap array event from early in the year, one from the middle of the growing year, and one from the end of the growing year. And so we can combine these species lists for an event to get the event diversity. That's actually supposed to read site diversity or trap diversity, but event diversity. So, so now we have kind of the species list for what's happening at a place. The metagenomic method is not really, not necessarily quantitative. It doesn't tell us how many individuals of each taxon are there or how much biomass of that taxon is there. And so to get at that, we're using high resolution imaging. We take a picture. Here's our setup. This is a 50 megapixel Canon camera, which ironically is out of focus in this image. But we use the diffuser sheets and a whole lot of light so that everything is bright enough and there are no shadows. This is an example of one of the images that's been downscaled here so that I can fit it into the PowerPoint. This image is stored as a TIFF that is about 120 megabytes. But the image that I've extracted to the right is the actual resolution and size of the image as we see it. So once we have all of the individuals in the image, we want to be able to extract each individual from the image. And so we use some software. It's an implementation of ImageJ called Fiji. And Fiji just stands for Fiji is just ImageJ. And so we are able to pull out for each individual in the image information about that individual. Once we have the images, we assign taxonomy, and we do this a couple of different ways. One of them is I look at it and I go, okay, that's a myriapod. I don't know myriapods very well, so I'm just going to call it a myriapod. But we can also use the species list taken from the sequencing to get a more refined taxonomy for the image. So I can look through the list of myriapods that were found for that event, and I can then use that to, and use images on the internet or field guides or whatever in the literature in order to assign a more refined taxonomy. So this is kind of doubling back, but another thing that we're able to do when we extract the individuals, we're able to extract traits for each one of those individuals. Now, the traits that we use are measures of body size and measures of shape. And I wrote a script that actually extracts the color of the individual in the image. So one of the graduate students associated with this project, Jarrett Blair, has been taking that trait data set and using it to develop machine learning tools to automate the identification of the images. So these are carabids that were collected by NEON and identified by NEON, so they were given to us with names intact. And so we imaged these beetles, we extracted their traits, and Jarrett is using these methods that he is able to identify carabids to species correctly more than 80% of the time using one of the methods. Now, moving forward with the invertebrate bycatch, which is a lot more diverse than carabid beetles, Jarrett is trying to develop methods that will allow us to identify things at least to order, if not family, across all of the invertebrates that we find in pitfall traps. But back to this, back to the pipeline. So in the end, I'm able to extract taxon abundance. We know the number of ants in the image. We're able to estimate the biomass from the measurements that have taken for the traits. And so for a given site, we know the abundance and the biomass and the diversity of the groups that we're studying. So for today's talk, we're going to talk about collections that were made in 2016. At each site, as I said, we did 3 trap events, 1 early, 1 mid, and 1 late, for a total of 42 trapping days per array. Now, you'll notice that we don't have much on the West Coast. It's just a little odd thing about NEON. In the rollout, the last places that they started were on the West Coast. And so we just recently got another loan from NEON. We got about 3,000 more 50 mil vials piled up in my office here. But it includes several trap years from Washington State and California. And we're able to more completely cover the continental US with samples. It also, some of these sites now, we have 5 trap years in hand for a single array. So the output of our metagenomics pipeline is not necessarily limited to the things that we can see or that we want to study. As I think it's important to point out that we are getting human DNA in most samples, and that's because these are not collected in a sterile fashion. They're pitfall traps collected in the field, taken back to the lab. Humans are interacting with it. But today we're going to focus on the 3 phyla that we're able to pull the metagenomics. We have some idea about them, but they're also large enough that we can actually look in the traps, in the samples, or in the images to back it up. So using those 49 trap arrays from 26 sites, all collected in 2016, and only focusing on those 3 phyla, the arthropods, the mollusks, and the annelids, we are able to get information from almost 300 families, 850 genera, and data set wide 1,424 species level entities. Now, the number of individuals in that study is actually taken from the images. And so what this is saying is that I had to look at 50,001 individual images and make some kind of statement about what I thought it was. Across the data set, there's almost 5,400 taxon by site occurrences. And so just to kind of quickly get at the stuff that my research focuses on, at all 4 taxonomic scales, we looked at species richness, genus richness, family richness, and order richness. At all 4 taxonomic scales, diversity is positively and significantly correlated with latitude. So this is the reverse of the standard expected latitudinal species gradient, where you should have higher species richness near the tropics and lower away from the tropics. I'm not going to get too far in the weeds, but I can say this is partially due to Alaska and Puerto Rico, but the relationship still exists if you remove Alaska and Puerto Rico from the analyses. So my research program focuses on kind of comparative biogeography. And I go about that a couple of different ways. One of them is to take the entire pattern, so the large scale pattern of arthropods and focal taxa. I guess I should also point out that arthropods they dominate the diversity of these traps. And so I don't plot them separately because they share the same pattern as the entire focal taxa pattern. But back to what I was saying, that I take the large scale pattern and then compare it to taxonomic groups within that large scale pattern to see how consistent they are with the overall pattern. And I also compare across taxa to see how consistent they are with each other. And so I took the 12 taxa, the 10 of the most abundant arthropods, and then annelids and mollusks, so that we'd be looking across 3 phyla. And once again, I'm not going to get too far into this other than to say, out of these 12 taxa, none of them showed the traditional latitudinal gradient and species richness, which would be a significant and negative relationship between diversity and latitude. And 7 of the 12 showed a significant reverse gradient in species richness. And now we will talk about the reason I was invited to talk to you all today. We took the species list from our metagenomics output, and for each species level determination, we compared it to the USGS Bison database, which stands for Biodiversity Information Serving Our Nation, which is a very smart way to name your database in the Trump era when this was generated. So just kind of taking a macro view of it all, non-native species richness on its own does not have a significant latitudinal gradient across the sites that we look here. That said, when you remove Alaska, which has very low non-native species richness, you do get a positive and significant richness gradient of non-native species. And if you remove Puerto Rico is depauperate in most of the taxa that we look at for this study. So if we're just looking at the continental United States, both native and non-native species show reverse and significant latitudinal gradients in species diversity. And just to point out a couple sites, I do find it so that the site with the most non-native species is the Smithsonian Conservation Biology Institute in Maryland. And note that Niwot in Colorado, which is a site that is alpine and tundra, has no non-native species that have been found in the pitfall traps. Now, if you take the diversity of the native species and compare that to the diversity of the non-native species, there is a positive relationship. So if you have more native species, on average, you also have more non-native species, which supports a contention by Dove-Sachs that non-native species do not necessarily decrease diversity. But if we go back to the complete data set and not just focus on the 3 focal taxa, Data set wide, there were 6,200 species by site combinations. Of those species by site combinations, 517, or just over 8%, were known to be not native to the region where they were found. So this represents 138 species from 7 phyla. And once again, they were found at 27 of the 28 sites because Niwot did not have any non-native species. Now, I do not work on introduced invasive non-native species in generally, so I'm not certain what is always the difference or if the differences are important amongst all the different groups. But if we just use the Bison database classification, it looks like we have 23 that are invasive and 58 that are known to be exotic. I think it's important to note here that for us to say that a species is non-native, our definition of a non-native species is that the Bison database tells us that it's non-native. And so this listing here is of the known non-native species that are in the Bison database. And to kind of put some pictures with this data, I looked yesterday and we did not find any emerald ash borers in the data, but I think that probably makes sense given their natural history that you probably don't collect them with pitfall traps very regularly. Some of these are well known. It seems to me that, so this slide was created by Cam, and I don't know if it's random, but it seems that a lot of these are pests of strawberries. But this is kind of giving a spatial understanding of where non-native things are being found. And I think that Cam only picked beetles for these slides. But it's important to note that, that our methods will allow us to detect and monitor for non-native species, but it also allows us to detect and monitor for native species. And so just kind of a quick and dirty, Cam went through and looked at each of the site lists to see how many family records. So these are state records where the family is not reported in the Bison Biodiversity database. So Bison has the non-native database, but they also have the distributional database for the United States. And so for each of these records that he's showing, he's saying that, so if you look at I guess I can use my arrow here, that if you look at the very top, he's saying that for CPER, which is the grassland in northeastern Colorado, Central Plains Ecological Research, something like that, that there was a phylum found there that was not recorded for the state. Remember that we're looking at all of the phyla, not just the 3 focal that we were talking about before, and that in this grassland, they found 2 families that had not been known from the state of Colorado. Importantly, so Cam has a graduate student named Sierra Smith, who, when I'm working with this data, I'm kind of bundling it at a macro level and not getting into the nitty gritty. Sierra went through the data, not completely, but went through a lot of it. And using the Bison range data, she compared each row with what was known about those data. So for the native species, she looked at 1,371 different occurrences of a species by site within a state. Of those nearly 1,400 occurrences, 770 of them were not known to have been in the state. There is no state record in the Bison database. It doesn't necessarily mean it's a old-fashioned state record, but it is something worth looking into. And about 600 were known to be found in the state. This is going back to the, just looking at the 3 focal taxa. Now, of those 1,300, about 1,100 were known to be in the United States. And 11 of them, I need to point out, that were known to be in Alaska. So they were in the United States, but they were not known to be in the continental United States. And so it's probably something that is found in the northern tier states of the US, but just had not been recorded, at least not in the Bison database. importantly, there's 200 species that we found that have no data in the Bison database. So we don't know if they're native or non-native. We don't know what their ranges are, but we do know that data doesn't exist in Bison for those ranges. Now, going to the non-native species that we found, Sierra looked at 200 site by species occurrences. About half of those were known to be in the state, and about half were not known to be in the state in which the site occurs. 72 of those, at least as far as the Bison database is concerned, were not known to be in the United States. And so these 72 species are of intense interest to us, that we need to dig in further to find out if it's just happenstance that Bison database does not have that data. And if we find out that they are not known, it's important that we are able to go back to the tubes and pull the vouchers. We still have the tubes here. Now we're using environmental DNA techniques where, for example, a herpetologist would go to a pond and filter the pond water to see if they can get DNA for the frogs that are in that pond, excuse me. But they, unless they make additional collections, they can't voucher the identification from the DNA with a voucher specimen. The sampling that we do, and it's helpful that we do non-destructive sampling, means that if that non-native species is in the sample, we're able to get it to the appropriate taxonomic experts and to I don't know, set off the alarms that there's more non-native species out there than we know about. I guess I should go back to the extraction phase of the DNA and point out that our lives would be made a lot easier if we could just pour the individual organisms into a wearing blender and grind them up. It would be much easier to get the DNA out of them than it is using the methods where we non-destructively sample. And so that's science-wise, that's kind of what I have for you today. But I almost always get questions about kind of the practicality of doing this. So we developed the techniques, then we got kicked out of the lab for a year and a half, which is good because it gave us time to analyze the data. But the way that we have been doing things, on average through the NEON samples, we get 10 traps per year from a given trap array. And doing this the first time, we used 10 filters. Each filter is like $5. And then including the chemicals, it costs about $27 per sample to go from the tube to the sequence data. This is not including labor. And so kind of sampling 10 traps per year, it's just under $300 per trap year, which I'm a field ecologist, so that seems to me to be kind of expensive. But we are doing experiments right now where we're actually taking the 10 trap years and doing half of the traps on one filter and the other half on another filter, kind of taking turns, even odds. And so we end up with 2 filters, greatly reducing the cost per sample or per trap year, because we're only having to run 2 samples of all the downstream events to get the sequence out. And so I just want to say thank you for listening. I hope that you all will see some of the utilities of the methods that we developed. We're very open about sharing, kind of have messy white papers of how to do what we're doing. And I want to point out that while these methods were developed with a particular trap design in mind, anything that is being stored in ethanol should work, provided you find the right primers. But if you're doing Malaise traps or Winklers or anything that is collecting arthropods into ethanol, I'm pretty certain that these methods could be effective for that. And that is all I have. I'm going to mute now and let somebody come in.
Well, thanks, Mike. This is interesting. This is kind of like, I get the idea that this is kind of like Zoom, you know, kind of what works behind it, and then pretty soon people are going to actually be like, hey, this information is really something we can use. So thank you for presenting this. I do have a couple of questions. Isabel says, how would these methods, and then in parentheses, HTS and imagery, compare to IDing and quantifying arthropods via morphology or with microscope? Faster, more accurate?
It's faster. I would say it's probably less accurate, but to a certain extent, it depends on what you're trying to get out. If you're kind of taking a macro perspective, a macroecological perspective, and you're worried about the abundance in biomass of ants, as opposed to the actual species identification for each ant, then it's quite effective. But so given the two slides that I have here, if you were to mount and pin every one of the ants on here, that looks to me, and I'm slow, but that looks to me like weeks of labor just to get it to the point that you can start to identify it. But given the species list, in many cases, especially for ants, given the species list, I'm able to assign species names relatively quickly, or at least genus names relatively quickly to the images.
All right. Jennifer asks, what portion of the samples were not identifiable, either not described species or could not be determined by the methods used?
So I can't give you a quantitative answer, but I do have, in my classification scheme, when I'm looking at the images, I have basically a taxon that I entered called no clue, that I don't know what it is at all. Other than that, I can generally assign things to higher classification. For most insects, I can do order, and for some orders, I can do families. But the stuff that I don't know what it is in the image, we just don't know. And I don't know how many of you have pitfall trapped, but when you pitfall trap, you get dirt, you get plant material, you get poop from things in the tubes. And so Neon does a reasonable job of cleaning the filth out, but sometimes they don't know what it is either when they're looking at it under the microscope. So it goes in the tube because it might be an organism.
Okay. Kevin asks, we collect in propylene glycol as well. How quickly do the samples have to move from the collection liquid into the preserving liquid, 95% alcohol, before you start to see degradation of the DNA, if you do?
I don't know. And we are getting some bad DNA. We looked closely at the temperature at which the collections were made. We were not worried about the post-collection transfer to the ethanol because I believe that occurs within 24 hours. There may be exceptions to that, but basically they go to the 10 arrays on a day, and then the next day they start transferring it to ethanol. We were concerned that the traps were out for 2 weeks in a warm, moist place like in Puerto Rico, or in a just blazing hot place like Arizona. We were scared of 2 things. 1, that the DNA was going to degrade, and 2, that the actual arthropods were going to melt. I've run pitfall traps in Panama where after 2 or 3 days, if it's raining and there's lateral flow and water gets in the trap, the arthropods are rotting before you actually collect the trap at 48 hours. And so we were very concerned about degradation of DNA and the organism. And so we looked at collection temperatures, has no effect on the amount of quality reads that we're getting out. We looked at precipitation to see if there was a dilution effect, and we see no relationship. We do have some sites that just for reasons that we don't yet understand, don't amplify as well. And we don't know if that's, something in the soil, something in the, I suspect one of the sites may not be using 95% ethanol, but I can't prove it. And so we've looked at everything that I think that we can to find biases due to degradation across space and time, and we just really haven't seen it. So that's not answering your question, but I don't know the answer to the actual question that you're asking.
Kevin says, thank you, that was helpful. We generally leave our traps out for 2 weeks and often get a lot of water, cutting our propylene glycol. Okay. Steve asks, where can the NEON survey results be found? Online in a database?
NEON survey meaning the results that we've produced?
Steve, you want to say yes, no? Which one you're looking for here?
Well, I can kind of start to answer that.
He says yes.
Okay, the results that we have are not yet publicly available in a formal way. I can email it to you, but in the long term, so NEON makes their collections, and then they send it all to a biorepository, which is at Arizona State University now. They just started it there a little while ago. The biorepository, I went out before the pandemic to meet with the people there. They have agreed to house, at very least, our high resolution images and the metadata associated with it. So that would be searchable by the site and the event that is in the images. It would also be searchable by the taxa that we find in the images. Now, the resolution of the image data, I guess it could also be searchable by the things found in the DNA. We just haven't discussed that yet. But if you had a taxon of interest or a favorite location that you think we might have studied, let me know and I'll send you what we found. And I'll certainly give access to any of the images if there's something that you're particularly looking for.
Okay, folks, I have just posted Mike's email in the chat pod. Steve, you might want to take note of that. It will also be in the email you receive tomorrow in case you'd like to email him and ask him any other questions, and so he can help you out with that. So I just thought I'd let you know that. Okay. Steve says, I have 70 carabid morphospecies from Pitfall Trap collections in watermelon from 10 locations in Indiana over 2 years. How might your results help me verify my taxonomic determinations?
I think the one, I assume that if it's a economic pest of importance, it would be in the database. I would not necessarily use the primers that we use to differentiate between closely related taxa, because one thing that I have noted in the NCBI database that we're using to get the taxonomy, I have some doubts that people that are uploading sequence always know exactly what they were looking at in the first place. And so SCBI taxonomy, so say I do a study, I'm doing a phylogeny of a group and I use molecular phylogenetic methods. I then upload the sequence to the database along with the name that I called it. Now that sequence is always going to be stuck with the name that I called it at that time. And so we all know that taxonomy changes over time, and we all also know that sometimes people make mistakes. And so if you had 10 or 20 taxa that are not closely related and you have suspicions of exactly what they are, then you could certainly use methods like ours to check the identifications. Not sure that was helpful.
Steve says thanks. I think at this point, that is the last question. And again, folks, you will be getting information tomorrow in an email from me that will also include his Mike's email and some other information. We are hoping that you will fill out the survey if you have a chance, so we can continue to bring these free webinars to you. And with that, thanks so much, Mike. We appreciate you taking the time and doing this. And this is something that I think is helpful for us, especially those of us dealing with things like invasive species. With that, folks, I'm going to end the meeting and have a good day from there on. Thanks again.
Thank you.